It's great to be here. I'd like to thank Jennifer for the excellent talk. I think that made a really great prelude to what I'll be talking about. So if we're thinking of the subject of epidemics and pathogen spread, I want to start with kind of going back 600 years and looking at spread of plague across Europe, where we see it takes all of, let's see which picked, it takes all of 4 years for plague to make it across Western Europe. But if we go forward to 2009, when swine flu emerges in Mexico in 2009, it makes it across the entire globe in just 4 months. And so because of our interconnected world, epidemics are more kind of troublesome and more fast spreading, but we have new tools to combat them. And so what I really want to focus on, and what Jennifer talked a lot about, is using sequencing to reconstruct pathogen spread. And I want to go into some kind of specific scenarios where this has been useful. But in general, what I'm thinking about here, and this is similar to the telephone game example, is this toy example where we have an epidemic that's occurring. So each red circle is an infection event that persists for a few days, spawns off a couple secondary infections. This one causes a couple secondary infections. And what we can do here is we can sample some of these individuals and sequence their infecting pathogens. So that's these blue dots. And then we can look at what mutations have occurred along this transmission chain. So we see that these 2 pathogens share this purple mutation, these 4 pathogens share this orange mutation, and we can use those mutations to reconstruct a phylogenetic tree. And so then from this phylogenetic tree, we can gain this from looking at our sequence data. This phylogenetic tree then tells us something about the underlying transmission process. It tells us that these 2 individuals were infected by a common ancestor, and these 2 by a common ancestor, and so forth. So these sorts of phylogenetic trees work on a variety of scales and also with a variety of different pathogens. So if we can zoom in and look at an outbreak of Middle Eastern MERS coronavirus in a hospital in Al-Hassa, we can zoom out and look at spread of Ebola across West Africa over the course of about a year and a half. Or we can zoom out further and look at the spread of flu across the globe over the course of about 12 years. So very generally, I think that this sort of phylogenetic tracking has the capacity to revolutionize epidemiology. It really takes a lot of kind of implementation, as Jennifer was discussing, but we just need to kind of smooth out the corners, and we're getting there. So in the talk, I want to really discuss 3 different things: Ebola spread in West Africa, Zika spread in the Americas, and how we can kind of pull this into the more real time. So first off, Ebola. This is going to be really talking about this paper that came out last year, led by Gytis Dudas, who's in the lab at Fred Hutch. And here we were looking at Ebola genomes that were produced over the course of the Ebola outbreak. So there were 1,600 genomes that were released. These were collected by different groups that were on the ground in different areas, sequencing infections. These 1,600 genomes were about 5% of known cases. And we were lucky in this case that this collection was actually well sampled in terms of space and time. So we didn't have one particular region that had too many genomes sequenced from it. So we're able to use this as a kind of a view into what's happening on the ground. And so we have then colors are showing cases in different areas, and circles are showing the kind of genome count. Sierra Leone, Guinea, Liberia, things are kind of well represented. So then when we have these 600 genomes, then we go and make a phylogenetic tree. So this is data from all 1,600. We have 1,600 dots here. So each of these is an individual Ebola infection. And the branching pattern that you see here comes from the genome sequence. So we can kind of know how these things are related based upon these mutations that have occurred over the course of the outbreak. We know there's a date range here that you can barely see going from 2014 to 2015. And what, in this case, we know the vertical kind of x-axis of when each individual infection was sampled. So we know this. And we also know where it was sampled from. So these are these different colors. So there's about 50 different shades of color here representing where these were collected from. And then it's this kind of job of statistical phylogenetics to reconstruct the ancestry here and reconstruct where, from a kind of a molecular clock, when ancestral viruses derive from, and also from these kind of phylogeographic models, where ancestral viruses derive from. And so we can actually, the kind of methodology here is advanced enough that we can do a good job at this and be able to kind of reconstruct this well. Our overall picture becomes that there is one introduction of Ebola into the human population. So there's a single spillover event from the animal reservoir. This happens in late 2013 into Guinea. It moves into Guinea into Sierra Leone, moves from Sierra Leone into Liberia, moves from Sierra Leone back into Guinea multiple times. However, we can also look in more detail here and look at each individual transmission event where the virus moves from one region within a country to another region. And so that's this picture here, where now this is showing each kind of arc is showing an individual transmission event. And we have been able to observe a lot of these migration events over the course of this phylogeny. And what we see is kind of emergence in Gekadu and Guinea, movement into eastern Sierra Leone, and then spread westwards. This color map is showing when the migration event occurred. So later migration events are kind of bouncing back and forth between Guinea and Sierra Leone before the epidemic dies out. And we can, now that we have this kind of list of migration events, we can look at what factors are associated with this. And so we do this kind of generalized linear model, and we see in this case that if there are two locations in the same country, we have more migration, as you might expect. We also get an effect where regions that are larger in population size tend to emit more migrants. And we also see that regions that are closer together just physically, physically tend to exchange more migrants. These two together form this kind of classic spatial epidemiology gravity model. So this kind of makes sense, but it's nice that we recovered it from the data. However, this effect of locations being the same country, even though it kind of might make sense, was actually an open question. So during the outbreak, it wasn't clear if the borders were completely porous. There were multiple border closures that had been implemented, but whether those were doing anything at all to slow migration, slow spread of the infection. And so what we see here is that there was an effect. If we look at two locations that share a border, there's about a twofold increase in migration rate within country versus between country, suggesting there was some effect on kind of slowing viral transmission by border closures. So we can also then look at this in maybe a bit more detail. So this is this exploded tree visualization that Gitas came up with. So this is the same tree as we were seeing here. It's the exact same data, but now what I'm showing is we're splitting it up so that every time there's a migration from one region to another, from one country to another, we split this out as a kind of as a clade. So what you see here is this is the initial introduction into Guinea, and it persists in Guinea for a while there. Then we have a single introduction from Guinea, from Gekadu and Guinea into Sierra Leone, from green into blue. And the remarkable thing that you see here is that almost the entirety of the Sierra Leone epidemic descends from that single introduction event, causes almost the entirety of the Sierra Leone epidemic. There's a few later introduction events that don't go very far at all. Almost the entirety of the Liberian epidemic comes from a single introduction event from Sierra Leone into Liberia. So in both these cases, suggesting that if we're able to kind of cut this transmission event and halt the transmission chain there, we could have averted a massive amount of kind of onward transmission. And so this kind of early warning situation. So this is at the country level. If we zoom in and look what's going on in Sierra Leone, however, within Sierra Leone and looking at each time we get a movement of virus from one region to another within Sierra Leone, we see there's much more kind of dynamic mixing in this case and much more kind of transient chains. So in this case, we see these kind of small clades that don't go very far at all. So these either die out or migrate elsewhere, and they only last for a few months at a time generally. We can kind of look at this further by, say, look within Port Loko and look across introductions into Port Loko, and we see that rather than there being a single introduction in Port Loko causing an epidemic within Port Loko, really we get about 20-odd different introductions that each have some small effect. So you think of when we kind of the classic, what the WHO situation reports were looking at, there'd be a kind of a time series of the epidemic in Port Loko, but rather than that actually being one kind of transmission chain, it's actually a number of kind of transmission chains that are stacked up on top of each other that are actually not related. And so dealing with these two situations is actually kind of different epidemiologically. And so then, overall, this gives this picture of really rapid spread of the virus, and these kind of migration events and the spread of the virus across the West African landscape is really driven by a large number of migration events that each by themselves don't do so well, but together kind of stack up to really push the virus and make it spread. Okay, so that's the overall picture for Ebola. I want to move on and talk about Zika. So Zika virus had these kind of sporadic but intense outbreaks within the South Pacific from 2012 to 2014, and then there was a very large epidemic within the Americas from mainly in 2016, very little in 2017. So I'm going to talk about a few different studies here. In the first one, this was a collaborative project that I was involved with colleagues from the UK and from Brazil, where the idea was to try to really figure out where and when the Zika epidemic started in the Americas, and really kind of hone in on that kind of historical basis. So this actually took the form of a bus trip, where we took a bus and drove it along the coast of northeast Brazil, stopping at these government diagnostic labs. They're called LACENs. So each lab had a kind of a state-level catchment area where samples were sent in. However, the diagnostic labs were generally not able to keep up with the number of samples being sent to them. So, so in the bus trip, we were able to kind of provide some very helpful diagnostic screening for these samples, identify which cases were positive for Zika, report that back to the LACEN, but then also take with us the positive samples. And then at the end of the bus trip, those positive samples were sent to local collaborators in Sao Paulo and Salvador to do actually the genome sequencing. So here's the bus. We have this kind of wonderful bus inception thing going on here. Here's me in the lab trying my best to help out. Here's the MinION sequencer that Jennifer was talking about. And what's been really great here is that earlier on in the Ebola epidemic, there was a lot of trouble with actually getting genome sequences produced, in large part due to the difficulties of getting samples removed from West Africa to elsewhere. But what's been much more effective more recently is actually just moving the sequencer to the samples rather than the samples to the sequencer. In just looking at kind of the epidemiological data, this is now just breaking up South America into different regions. Here showing a time series of case reports. So these are not diagnostically confirmed here. These are just notified cases. And we see already that something's looking kind of special in terms of Northeast Brazil, where Northeast Brazil has this epidemic peak, a smaller peak, in June, July, August in 2015 that doesn't show up elsewhere. We don't see that epidemic peak in other places. We were able to actually confirm these through our diagnostics, through our RT-PCR, that yes, this peak was indeed Zika virus. So this is suggesting at this point that Zika was in Northeast Brazil before it was elsewhere. But we're able actually to kind of strongly confirm this via genome sequencing. So this is showing a phylogenetic tree like the Ebola tree, where it's time resolved and space resolved. And what you can see here is there's an outbreak in French Polynesia in late 2013, early 2014, and that all of the genomes that we've been able to collect from South America, and then hence the millions and millions of cases, all of those cases descend from a single introduction event from French Polynesia, from the South Pacific, into the Americas that arrived in early 2014. We think that it arrived in early 2014 due to kind of this molecular clock dating, but we can also place it in Northeast Brazil based upon the phylogeny, where you can see the structure generally where the blue branches, which are Northeast Brazil, are outgroup to other viruses, and that other locations kind of nest within diversity that's occurring in Northeast Brazil. So you can kind of see that visually. The statistical model places that 90% confidence that this branch, this node is in Northeast Brazil, 99% confidence that this node is in Northeast Brazil. And so we get this picture then that there's a single arrival event in early 2014 and then spread from Northeast Brazil. But the generally remarkable thing here is that this here in June 2015 was the first detected event of Zika in the Americas. So we now have strong evidence that Zika was circulating for a full year and a half in the Americas before it was even noticed. And there was this huge amount of cryptic transmission. And by the time it was noticed to be circulating, it had already got into Central America, to the Caribbean, elsewhere in Brazil, and had really circulated widely. Okay, so that's the kind of the big picture thing. Looking at a more kind of detailed analysis here, this is a collaboration with primarily folks at Scripps, Nathan Gruba, Christian Anderson, but a host of other people as well, looking at Zika spread within Florida, so a much smaller scale. So during the course of 2016, when there was the huge epidemic in South America, there was actually thousands of Zika cases in the United States. However, if you follow up most of those cases, you'd find that people had recently visited Zika-affected areas. However, the Florida Department of Health noticed in Miami a cluster of 250 cases where those people had no history of travel. And so those were assigned as locally acquired infections just based upon interview. They were able to then, the Department of Health kind of identified 3 different transmission zones in Miami, Little River, Wynwood, and Miami Beach, where Zika was circulating. When they identified this, they then ramped up vector control, and then these black lines are showing mosquito abundance drops, and the epidemic or the outbreak appears to be kind of averted and brought down as well. And then what we're able to do is to look at sequences from the genome sequences from these viruses, and then reconstruct what resulted in that outbreak. So this is a similar picture as we were just looking at, where it gets spread from French Polynesia into Brazil, from Brazil into the Caribbean, and then we see going from the Caribbean into Miami. And this is now a blow up of this section here, where we have outgrouping to the Miami outbreak is Dominican Republic in orange here, Dominican Republic in orange here. So it looks like there's 2 separate introductions into Miami that each do well. They arrive around March and then spread and aren't really noticed until July. And then also kind of helpful is that there was a number of mosquito pools that were sequenced. And what we see is that the mosquito viruses, the viruses that are present in mosquitoes, are interdigitated with viruses that are present in humans, suggesting kind of as expected, a human to mosquito to human transmission chain and not very much sexual transmission, which actually had been a question. So we see that there's this tight connection to the Caribbean. If we just look at the number of infected travelers that are arriving, our estimated number of infected travelers that are arriving, we see that the Caribbean, mainly through actually cruises, is contributing a lot of what we expect to be Zika arrivals. So it makes sense that we see that the kind of actual cases derive from the Caribbean. This is some modeling work that I did where we see kind of 2 big clusters of virus rather than a lot of single kind of one-off introductions. This has information on how well the virus has been spreading in Miami, and this is able to kind of place the R0, which is this kind of fundamental number for viral spread, between 0.4 and 0.8, which is somewhat helpful. And now we're kind of trying to recapitulate the study. This is led by Ali Black, graduate student Ali Black in the lab, where we've kind of jumped on board the MinION bandwagon. Ali went down to the USVI a year and 3 months ago, collected virus, so then did some sequencing on island and some sequencing at the Fred Hutch. We now have 31 genomes from the USVI, and have identified a large number of introductions that then went on to circulate in the US Virgin Islands. So generally, I'm hoping that this was somewhat convincing, and Jennifer's talk especially, somewhat convincing that these sorts of inferences can be helpful, and that we can now kind of try to make them more actionable. The difficulty is that in this case of, say, Zika in Miami, though that outbreak occurred in summer 2016, the paper was published in May 2017. If we look here at these Ebola studies, Tong et al. Samples were collected in fall 2015, data released in May 2014, data released in May 2015, paper published in June 2015, other papers also with this roughly year-long gap. So generally, there's been this very large discrepancy between when samples are collected and when kind of genome information, when inferences become available. So more recently, we've been trying to work on this. And so here, in kind of trying to think about this problem, there's some key challenges. Namely, the biggest one is that timely analysis and sharing these results is critical. We just need to be very fast here. The dissemination must be scalable, so we can't rely on emailing PDFs back and forth. We have to be able to integrate across many different data sources and data formats, and the results must be easily interpretable and queryable by people who are not experts in phylogenetics. And so what we came up with is this next strain project. This is all a close collaboration with Richard Nair at the University of Basel. James Hadfield here in the audience has been instrumental in kind of making this work well. Other people who have been involved are Colin, Sydney, John, Barney, and Charlton. So generally the architecture is set up. Oh, I should say that first off, that all the code is open source. It's all up in GitHub. Everything is there. You're free to use, download. We're trying to do this all in an open fashion as much as possible. So basically, there are 3 components. There's a database written in Rethink, where we pull in data from a variety of sources, from GenBank, Virological, GitHub, et cetera. And when we pull into the database, we do our best job to clean it up, fix things like date formats and country names and all of these little kind of idiosyncrasies that always exist, clean it up into kind of clean data in our database. Then we do an export from that database to Augur, which is our Python informatic processing scripts. This is doing things like aligning sequences to our reference genome, building a tree, estimating a molecular clock, estimating geographic spread, et cetera. So all of our kind of models here. We're happy with this in that we can process the 1,600 Ebola genomes in just 2 hours, making it something that you could actually run this day to day as new data arrives. And then when this processing is finished, we kind of export this bundle for visualization. This gets pushed up to the internet to this front end called Auspice, which is written in JavaScript. But so really what you interact with, what the user interacts with, is a website, is nextstrain.org, which you can see on your laptops now or phones, but I'm going to give a little demo. So the landing page is a number of different viruses that we're tracking. At the moment, Zika, Ebola, dengue, seasonal flu, avian flu, mumps, measles. If we look at any individual, so say Zika here, what we have in kind of general format is a sidebar on the left, and these are kind of all the different things you can manipulate in the app. And on the right are different kind of visualizations. Primary among these is a phylogenetic tree that shows how viruses are connected, and a map showing how virus genomes are connected, and then a map showing how viruses are connected in physical space, and also a view of the genome showing which mutations have occurred and which sites are more variable. And so what you can see here is we've kind of tied this all together. So if we say manipulate time, rewind time, and come back to early 2014, we can see that early on, viruses appearing here in Guinea in green, and then showing up here in the map in green. And then as we, this is Ebola. And as we move forward in time, we see virus spreading and emerging from Guinea into Sierra Leone, spread from Sierra Leone into Liberia, and so forth. We can look at this in a more kind of dynamical fashion by playing this animation, where we kind of just come along time, and as we basically scroll through the tree, you can see spread of virus and how it takes off in different areas, making kind of this dynamic visualization of viral spread. I'm going to stop this. We've tied this all together, so we can look at, say, if we change the coloring, we can look at, say, date, and see that in this case, you can see very clearly that earlier viruses are here in Gekadu, and then later viruses are occurring in this other region here. We can also look at individual mutations. So there's this very classic mutation that was later shown with functional work to be related to kind of how well the virus is doing in cell culture. And in this case, we can kind of see how this maps to the tree and see which individual branches have mutations to them. We can also do simple things like look at different views of the phylogeny. This unrooted tree is more useful for seeing what actually is genetically similar versus what is kind of spreading. We also can see kind of how fast Ebola is evolving based upon kind of this molecular clock. And we can do kind of a number of other things. One thing that you can imagine being very useful is this kind of contact tracing. So you could imagine kind of field epidemiologist being interested in what's happening in this particular clade of virus. And you can see then these viruses are very closely related to one another, and they're in this just very little pocket in between Sierra Leone and Guinea, and it's something then that you could imagine actually looking to see, where did it go? Actually looking to see how these viruses how these individual cases are connected. So that's roughly Ebola. For Zika, we get a kind of a similar picture where we see spread of virus from French Polynesia into the Americas. We can kind of, again, rewind time, see spread from French Polynesia in Brazil, Brazil to the Caribbean, to Central America and onwards. We can also do things like if you are, say, Ali in the lab, interested in how your data fits in with things, we can look at these particular genomes and just look at these genomes from the USVI, see how they connect to other things. Or we might be interested in what's happening in Brazil, and then we can subsample to just Brazil and see how Brazilian isolates connect elsewhere. So there's other features here, but I don't think I have time to go into them. So generally, this has been fun to work on. It's been nice that this has been getting some press, which has been fun. But kind of even more gratifying is that this has actually been getting some use. So that these are colleagues in Sierra Leone near the tail end of the Ebola epidemic. They were sequencing viruses, emailing us, and then we were getting them onto the website and getting them updated so they could see kind of exactly how their viruses fit in with other things that were circulating. Generally, the desired analytics are pathogen-specific and tied to outbreak response measures. So in another example here, related to Jeff's talk earlier, we have seasonal flu here, and we're trying to do kind of predictions of which strains will be circulating. And we're working in for flu, it's really kind of what strains are circulating rather than where it's circulating, because for flu, the response measure is how do we make a better vaccine and which strains we put in the vaccine, whereas for Zika, it's going to be really where should we target our vector control. For Ebola, it might be something like contact tracing. And so these measures are very much pathogen-specific, though generally, we're working to build out pipelines for different pathogens. We're trying to improve our databasing to make it easier to kind of share data with us, and we're trying to lower the bioinformatics bar so that people who aren't us can do these analyses more easily. And with that, I'd like to close. Thanks, folks in the lab. Thank Gitas and Andrew for the Ebola work, Nick, Nuno, Ollie, Nathan, Ali for the Zika work, Richard and James for the next strain work, and happy to answer any questions. Thank you.
Thank you. Is there any questions from the audience? We are ahead of time, 5 minutes. So then I have one question. So you trace the viruses by point mutations mostly, right?
Yes.
Is that all neutral evolution, or is there evidence for adaptive selection?
So to look at a couple examples here, it's thought to be the case that, so in the case of, say, Ebola, almost everything is neutral. There were kind of maybe a couple examples where it might have been non-neutral. In the case of Ebola, it's almost completely neutral evolution. And the patterns that you see are really driven by epidemiology of kind of which lucky virus happened to arrive in a susceptible population. Whereas flu, there's much more positive selection. And what that manifests as generally is these trees. I can give a better example here. These trees where, color by antigenic advancement. So this is showing kind of how different a virus is in terms of immune system. And what you can see as we go from blue to green virus, that the kind of derived viruses really take off and do well. And so you get this very characteristic pattern where there's a particular strain of virus that does really well and other strains of virus that die out. And we can kind of relate this to particular mutations, and those are abrogate antibody binding and so forth. So there appears to be generally a widespread in terms of which viruses are really undergoing a lot of positive selection and which viruses are more neutrally evolving.